on this computer. Okay, so I'm gonna start my class while we are waiting for your friends. There's still maybe three more people to join. So the class today, I'm gonna give a lecture. So I'm gonna introduce myself again. So I'm Ajahn Anandi Chanon. So today I'm gonna teach you about the adaptive immunity. Before that, I'm gonna give you guys I'm gonna recap the lesson that you have learned during the introduction and also the innate immunity. Talking about the immunity, you guys need to understand that right now we separate the immunity lastly, I would say that just lastly by innate immunity and also adaptive immunity. When we compare between the innate immunity and adaptive immunity, we said that so all of them, both innate immunity and also adaptive immunity, both of them need to discriminate. The discrimination is mean they need to know what is a foreigner, what is yourself, and the immunity should attack only foreigner, not attack yourself, except when you guys got the damage or abnormality, you may attack yourself. But anyway, we try to keep our body healthy. And when we compare between the innate and adaptive immunity, the adaptive immunity is going to be a highly specificity when compared to the innate. So the adaptive immunity is mean that one clone, it needs to specific just only one thing. We call this is a clonal specificity. And also one clone, even they bind to one antigen, they also have a lot of antibody or even a lot of receptor to bind. The effect that a lot of receptor or a lot of antibody that can bind on one antigen, we call the diversity. And even your body, you have a lot of antibody, you have a lot of receptor, and you can bind to a lot of things in the world. We call this effect as the diversity. But if it in the innate gonna remember only the pattern. If you can remember the innate immunity, they just remember the pattern of something. For example, the pattern of bacteria, the pattern of the fungi, or even the pattern of the damaged cell. So the innate one just remember the pattern, but the adaptive immunity, they remember as the clonal. One clone remember just one thing. So the adaptive immunity gonna be a lot of diversity. And also the innate immunity, they just prepare before you're born. And even when you're born, it need to be ready to use. And it need to be immediately used. So there is almost no any improvement, but compared to the adaptive immunity need to improve themselves from time to time. It mean that when you was born, you have a very, very low adaptive immunity. But when you are growing, and growing, you're gonna adaptive. You're gonna improve your immune from time to time. So this is the capacity of the adaptive immunity. So the most important thing, the innate immunity, they just respond to everything, just remember by the pattern. There is no any memory. But for the adaptive immunity, there is a memory. So they need to memorize something. And when they got the same thing, when they got infection by the same thing for the next time, it's gonna be faster and even more effective. So this is a feature of the adaptive immunity. So when we're talking about adaptive immunity gonna divide by two mechanisms. Lastly, it's just lastly. First thing, the adaptive immunity gonna respond by using the cell-mediated immune response or the CMIR. The cell-mediated immune response means that you need to respond by using the cell. So talking by using the cell, there is two cells. You need to imagine there is only two cells need to go out at the site of infection. So there is a T helper cell. So the T helper cell, they're gonna secrete cytokines. So if you want to remember the marker, it's going to be CD3 positive and CD4 positive. So this is the marker of the T helper cells. And if you got the intracellular pathogen or even yourself got some abnormality, for example, you got a tumor, you need the cytotoxic T cell need to attack the abnormal cells. So the cytotoxic T cell gonna attack to the infected cell or even a tumor cell. So the marker of the cytotoxic T cell is CD3 positive and CD8 positive. So for the cell-mediated, it need to move to the site of infection and do function. But if we're looking at the humoral immune response or the HMIR or the HIR, the humoral immune response is gonna be antibody. So you need to imagine the B cell, they don't need to go out. They just stay at home and then they just secrete the antibodies. So the HMIR or the humoral immune response, they just secrete antibody by the B cell to attack to the pathogens or even to neutralize the virus or even the toxin. So if you want to remember what is the marker of the B cell, it is CD19 positive. So this is just only the recap, okay? So the helper T cell, we say that, okay, it's just existing and then they want to enhance immunity by using the cytokine. So the cytotoxic T cell that just specifically attack the abnormal antigen on the surface. So if you have any abnormalities, it's gonna attack that cell. And the last one, B cell, is just secrete antibody. So this is the recap of the basic knowledge that you need to know before come to my class. And then if you want to know the T cell, have a lot of subclass. So the T cell we have the T helper 1, T helper 2, and also today, nowadays, we have T helper 17 and also the T follicular helper cells, the TFH. So the first one, the T helper 1, the key cytokine, the T helper 1 gonna secrete interferon gamma, and this interferon gamma gonna help macrophage to kill the intracellular pathogen. So we said that the T helper 1 is the very key player for the enhancement of overall bacterial killing, especially intracellular bacteria. And then if we look at the T helper 2, so the T helper 2 gonna secrete IL-4, 5, and 13. I would say that if you can't remember that, you just know IL-4, that's it. So the IL-4 gonna enhance or gonna activate the eosinophil, and also sometimes it's gonna activate the mast cell to do the degranulation. The degranulation is mean that this cell gonna secrete the granule outside and do something on the humans. So the parasite gonna be attacked by the granule from eosinophil and even from the mast cell. So we say that. So the T helper 2 is an enhancement defense against the parasite. And moreover, the T helper 2 also have the function about the wound healing as well. So it means that you need to imagine when you got bacterial infections, you have a very, very high response of the T helper 1. And then if you get rid of infection and you want to heal your body, you're gonna have the T helper 2 respond later when you got healing. In fact, in the real situation, I would say that T helper 1 gonna increase and then T helper 2 gonna increase later. So this is a counterbalance. If you have the balance response between T helper 1 and T helper 2, I would say that you're gonna stay healthy. And then if you got infection by some extracellular pathogen, and also if you got the extracellular pathogen infection or even you got a fungi, I would say that under your skin or even your intestine or even in your lung, if you got infection, you need to call the neutrophil to the site of infection. The T cell have a special cells right now we call this cell as a T helper 17 because it can secrete the IL-17. This T helper 17 normally they stay inside our tissue, and when you got infection, IL-17 gonna call the neutrophil from the blood and go to the site of infection. So this T helper 17 is very, very important against the mucosal response or even to call the neutrophil to the site of infection. And the last one, not actually the last one, there is a T cell, the helper cell that can enhance immunity. We call the T follicular helper cell or the TFH. The TFH is very important because it can secrete the IL-21. IL-21 is very important cytokine to activate the B cell. I would say that in my last part of my lecture, I'm gonna go into the detail of the TFH on doing on the B cell. This is very important. If we have no any TFH, B cell cannot have a cross switching, cannot have any memory, and cannot have any adaptive response. They gonna secrete only IgM at the end. So this is just a recap. All of them, I just would like to remind you what is the knowledge that you have learned from the introduction and even from the innate immunity. So today, my objective of my teaching today, I would like you to know how the adaptive immunity could be produced, and after the produce, they need to mature. This is the thing that different from the innate immunity. So the innate immunity is just produced and then it's ready to use. But for the adaptive immunity, they need to produce and then they need to mature before and to be ready to use after maturation. So what is the mechanism behind that? This is the first objective. And then you need to know when the adaptive immunity has been matured and also have been stored for some side of your body, how could the T cell could be activated. And then I'm gonna teach you how the B cell could be activated. And then if you know about the T cell activation, B cell activation, you need to know how the B cell could have a selection and affinity maturation, because as I told you that objective number 4 here, need the working together between T cell and B cell and working together. And last one, for the phase of exposure, you need to understand this is the adaptive immunity. The first time you encounter the antigen, the response gonna be very slow, but after you expose it again as a secondary exposure, your response gonna be faster. So I'm gonna give you a lecture about this. So come to the first objective. The first objective is mean how could you produce the adaptive immune cell. So this slide, I also saw this slide in Ajahn Visit's lecture. So you need to understand all the white blood cell or even red blood cell or even the platelet cell, all the cell in your blood could be not all the cells in your blood was produced by the bone marrow. But for the adaptive immunity, we will focus only the T cell and B cell. We call these lymphocytes as a lymphoid. So all these things are the lymphocyte. The different thing from the innate immunity is that when you produce the B cell, I'm gonna start from the B cell first because the B cell is easy and also very simple. Start from the B cell. These stem cell on in the bone marrow, when they differentiate themselves, they're gonna go and come to the B cell lineage. So the B cell need to expose to your self antigen as a selection. Any B cell that could recognize or could bind to your self antigen need to be apoptosis. So it means that this is a negative selection. Any cell that could bind to your antigen need to be die. Because if you have some cell that can recognize or can bind to your cell, you can produce the autoimmunity. So the first step, you need not to recognize or need not to bind to your self antigen. So, but there is a second chance. So if this B cell could bind to the self antigen, there is a second chance. So the B cell gonna allow to have a mechanism as a receptor editing. So it means that they just take back the receptor that can bind to your self antigen. It's just take it back and then change the receptor. Look at this. At the first time, it's like a circle here, the half circle receptor, but it could bind to the antigen. So this receptor have been taken back, and then it's generate a new receptor at the triangle right there. So the triangle receptor could not bind to your self antigen. This cell gonna be mature. So we select just only the B cell, not self-reactive. If any B cell got a self-reactive, it could be go to the receptor editing. We call this mechanism as a negative selection. Those are about the B cell. So about the T cell, so the T cell will be produced by your bone marrow. But, oh, sorry, the B cell, the B is stand from the bone marrow. It means that the B cell will be produced by the bone marrow and mature to be ready to use at the bone marrow. That's why we call the B cell. For the T cell produced by the bone marrow, but it's just immature T cell. So the T cell, the immature T cell, gonna transfer, then it's gonna move to the thymus. That's why we call T cells, because even it produced by the bone marrow, but it go to the thymus for the selection, for the maturation, and to be ready to use at the thymus. So the T stand for thymus. We call this is a T cell. So the step of the selection is starting from T cell gonna expose to your self antigen. So if T cell bind to your self antigen so high affinity, is so strong, the binding is so strong, it need to be apoptosis. But it is very important, and this is very special. If the T cell could not bind to your self antigen at all, could not bind at all, it also need to be die at the end, because your T cell need to have the receptor, some part of the receptor need to bind to the MHC molecule. I'm gonna have a picture later that, but anyway, for the T cell, it need to weakly self-reactive. So why we need to weakly self-reactive? Because we want the T cell receptor bind to the self antigen. I would say that in my slide, if you have the brain signs like this, the brain sign like this, I would say that you need to go back and you need to relearn. And also my examination gonna ask you about this thing, because this is very important. So the selection of the maturation of the T cell and B cell is very important. If you fail anything, if something wrong, you're gonna get a disease. So the B cell should be not self-reactive at all. But for the T cell, it needs to be weakly self-reactive. So this is a picture. If you look at this, so this is a B cell receptor. I would say that when we call the B cell receptor, it's actually antibody. The B cell receptor is just a surface antibody bound on the membrane of the B cell. But if it secrete out, we're gonna say this is an antibody. So the antibody on the B cell, or even the antibody that secrete out, we also call the B cell receptor. So the antibody on the B cell, we call the B cell receptor. If you look at this, so the B cell, all of the receptor bind on the antigen. But if you look carefully, so the T cell, if you look carefully, there is a two-step of the T cell receptor. So the T cell receptor, in principle, T cell receptor recognize to the peptide presented on the MHC class 2. This is the reason why our T cell receptor need to recognize to our self antigen weakly, because we need to weakly bind to the MSC molecule, and we have the second half here to bind to the peptide. So it means that the T cell receptor bind two things at the same time, weakly bind to the MSC molecule, and then bind to the antigen at the second step here. And then after the T cell and B cell going to the mature state, I mean the B cell maturation in the bone marrow and also the thymus, the T cell mature in the thymus, or T cell and B cell gonna move to the peripheral lymphoid organ. So the site of generation, the site of production, we call the generative lymphoid organ, or the primary lymphoid organ. Any word is okay. So the T cell and B cell is gonna stay in the peripheral lymphoid organs, or even we call the secondary lymphoid organs. So it could be stay in the lymph node, in the spleen, or even sometimes they could stay in the mucosal lymphoid tissue. So talking about this, the generative or primary or central lymphoid organ, all these things are the same word. So you just use any word is okay. So the generative primary or central lymphoid organ, the function is just produce white blood cell. But the B cell maturation in the bone marrow, T cell maturation in the thymus, that's it. So the important thing after the maturation, T cell and B cell need to go to the secondary lymphoid organ and wait for activation and development. This way, where they stay, gonna have the different function. So if they stay in the spleen, that the structure of the spleen, there is a lot of vein and also artery. Look at this picture. There is a lot of vein here and also a lot of artery. So it means that the T cell and B cell in the lymph in the spleen, they stay and also they are the filter for antigen come with the blood. So it means that if you have any infection, if you got any blood antigen inside the blood, your spleen gonna activate T cell and B cell inside the spleen. And if the T cell and B cells stay in the lymph node, you just need to imagine. I would say that all throughout your body there is a lymph node stay throughout your body, scattering throughout your body. So if the lymph node has a T cell and B cell, those T cell and B cell gonna be a surveillance of the cutaneous antigen. Look at this picture. You can see here that there is a white circle here, white knob here, white knob here, white knob here, white knob here, a lot throughout your body. All these things are your lymph node. So if you got infection on your arms, I would say the first activation site gonna be lymph node at your arm. If you got infection at your leg, the first activation site of your leg is gonna be a lymph node at your leg. So this is the surveillance at your cutaneous antigen. And also there is a principle of the T cell and B cell stay in the mucosal lymphoid tissue, or sometimes we call the MALT. So the mucosal associated lymphoid tissue, it means that if you look at here, this is a structure of your intestine. Under your intestine, there is a group of lymph nodes like this. There's a lot of T cell and B cells here. So this cell gonna have a function we call the gut associated lymphoid tissue, or the GALT in your intestine. There is a function to stay and also survey for the antigen of your intestine. It means that when you eat something and those things are very dangerous to your body, you're gonna activate the immune response in your gut associated lymphoid tissue, or sometimes we call GALT. Anyway, you need just to remember that the cell produced at the central or the primary or the generative lymphoid organ. So the T cell and B cell produced by the bone marrow. B cell say maturation in the bone marrow, why we call B cell. But the T cell produced from bone marrow mature in the thymus, that's why we call T cell. So the secondary lymphoid organ, so the spleen, if the T cell, B cell go to the spleen, the function is for survey antigen in your blood. If it go to lymph node, it just survey antigen for your cutaneous or for your skin. If they go to the mucosal lymphoid tissue or go to the gut, it's also survey for the antigen to your intestine or to your lung. That's it. That about the first objective, and then I'm gonna go to my second objective about the activation. Right now, you need to imagine you produce the T cell and you produce the B cell. They move to your lymph node or they move to the spleen. They just stay still, do nothing until antigen comes. So you need to imagine here, you're starting from, you got some pathogen infect to your skin. For example, it infect to your skin. I would say that T cell and B cell never ever go out and do function at the site of infection, never ever. But when you got infection, the cell, the dendritic cell, gonna take some antigen. Dendritic cell takes some antigen, and then this dendritic cell from the site of infection gonna move to the nearby lymph node. This dendritic cell gonna present antigen to T cell or even for B cell. So present to the T cell gonna develop themselves into the T helper cell, to the cytotoxic T cell and whatever. And then it's gonna produce the effector cell here. only thing that moved out from secondary lymphoid organ to the site of infection is just the effector cell. Again, I'm gonna tell you again. So T cell and B cell, after they produce and mature, they gonna stay still in the lymph node and wait for activation. And then when the dendritic cell survey and got some antigen, they're gonna collect antigen and move to the lymph node nearby. And the dendritic cell gonna present antigen to naive T cells. Present to the naive T cell, and then these naive T cell, after got the presentation, they're gonna develop into the effector cells. So only the effector cell gonna move out and go to the site of infection to do function. Now I gonna focus on the mechanism of the antigen presentation. So this is the thing that happened. Once the dendritic cell eat some antigen, the dendritic cell need to digest. This antigen protein, they have three epitopes here, the pink one, blue one, and brown one here. They need to digest. And once it digests, we call the digestion here like a antigen processing. Once this antigen has been digested, the dendritic cell need to present the peptide, the short peptide, by putting this peptide on MHC molecule class 2, and then the specific T cell that bind to both peptide and MHC class 2 here, the specific cell gonna bind to this dendritic cell and go into the activation. I would say that this is come to the problem because when you eat one protein, there's several epitopes could be occur. So the dendritic cell need to select just only one epitope. So any epitope here that can be bind to the MSC class 1, class 2 most effectively, any epitope with the most effective binding with the MSC class 1, class 2, we call these epitope as an immunodominant epitopes. Any epitope that can bind to the MSC class 2 most effectively, we call immunodominant epitope. So right now, you need to understand, once the dendritic cell presents the antigen to the T cell or to the naive CD4 T cell, this is the thing that happened. So the dendritic cell present peptide or the MSC class 2 and then bind to the T cell receptor. There is a first signal come by this activation. So once this signal activate, this T cell gonna produce the CD40 ligand. This 40 ligand, CD40 ligand, gonna bind with the CD40 or the dendritic cell and lead to the lead to the binding glue. So it means that when the dendritic cell present antigen to the T cell, it could be move out from each other. So the T cell need to express CD40 ligand and bind to CD40 on the dendritic cell to make it move closer and close to each other. And then the T cell and B cell, the dendritic cell need to explain the B7 here by to the CD28. And this we're gonna call the secondary signals. To activate the T cell, I would say that to activate the T cell, you need both first signal and the second signal. You can't lose anything to effectively activate the T cell. So if the activation occur on the CD4 cells, you're gonna get helper cell at the end. If the activation occur with the CD8 T cell, you're gonna get cytotoxic T cell at the end. So the function of the helper cell is just secrete cytokine, just like I told you at the beginning. But the function of the CD8 T cell, cytotoxic T cell, is about killing of infected cell or tumor cells. This is the application. That I told you that when you activate the T cell, you need both signal of the first signal here by the MSC class 2 with peptide and also with the TCR. And also you need the B7 and CD28 each other. So the first signal here and also the secondary signal here, you need both of them. But if you don't want to activate, the T cell, you just use the analog here. The CTLA4, this is the molecule that got the Nobel Prize. This is the Nobel Prize molecule. The CTLA4 can inhibit the activation of the T cell. So if you inhibit the activation of the T cell, you're gonna have no any T cell. So it means that you have no any killing abnormal cell in your body. We call this the no response anything. We call this is energy. It means that it means no. Energy is mean energy. So the energy to activate the T cell is not enough. We call this effect as energy. The important is mean that if you want to have any organ transplantation, if you want to give your liver or if you want to give your kidney to people, if you directly give your kidney to people, I would say that your kidney gonna be destroyed. The only way to prevent the destruction of the organ transplantation, you need to inhibit the T cell activation. So this is the example of the interference of the antigen presentation. So this is a break, just like I said. Okay, and then right now, once you activate the T cell into the effector cell, I mean the helper cell or even the cytotoxic cell, it need to go out and move from the lymph node and go to the site of infection and do their function. So this is a function of the CD4 and CD8. So if the antigen presentation, if the peptide present by using the class 1 MSC, if the antigen presentation present by using the class 1 MSC, is you're gonna activate the CD8 T cell, and then you're gonna have the cytotoxic T cell. The function is all about the killing of intracellular infected bacterial cell, or even you're gonna kill the virus infected cell, or even you're gonna kill the cancer and tumor. So this is all about the killing abnormal cells. But if the antigen presentation presents the class 2 MSC, I mean the peptide present on the class 2 MSC and present to the CD4 T cell, you're gonna get the helper T cells. The helper T cell is very important to activate the macrophage to kill the intracellular microbes. And even the same thing, but the B cell also have the function of the presentation as well. So the B cell can use the B cell receptor bind to antigen and then taking to the B cell and then present the peptide antigen on the MSC class 2 and then activate the T cell into the T helper cell, and then also the T helper cell gonna activate back. The B cell could go into the plasma cell and secrete antibody at the end. So this is the phase of activation. I'm gonna remind you again that when you got any infection, for example, this is a tissue of the infection. So when the microbe infect your skin, that is gonna have some protein secrete from the microbe. The dendritic cell gonna take that antigen and then go out from the site of infection and move to the nearby lymph node. Come to the T cell, the naive T cell. So the dendritic cell gonna present, for example, if the T cell is a naive CD8 T cell, this dendritic cell gonna present the peptide on MCLC class 1 and also have the co-stimulation as a second signal. And then this naive cell gonna turn to the effector cell. Once you got the effector cell, the effector T cell, I mean the cytotoxic T cell here, gonna move out from the lymph node and go to the site of infection and kill the infected cell. This is a mechanism. And also about the killing. So this is an infected cell. So the first effector cell of the cytotoxic T cell is mean that the cytotoxic gonna use their receptor bind on the infected cell because the infected cell gonna have the peptide present on the MHC class 1. The mechanism is mean that the T cell, the cytotoxic T cell gonna secrete the perforin. The perforin has a function to make a pore. So this is perforin make a pore on the infected cell. And then the granzyme also gonna be secreted and the granzyme gonna go to the infected cell and induce infected cell to the apoptosis. So this is the first mechanism by using the perforin granzyme mediated killing. So at the end, it's gonna be to the apoptosis stage. The another mechanism is the T cell, the cytotoxic T cell, once they recognize to the infected cell, the T cell gonna express the fast ligand bind to the fast. So the fast receptor bind to the fast ligand on the T cell here is gonna activate the cell into the apoptosis. So this is the inside detail, but I would say that you don't need to remember. So right now, come to my almost the end of the T cell lecture. I'm gonna give you the take-home message of the T cell. So the T cell they produce from the bone marrow. The bone marrow, you're gonna get only immature T cell. The immature T cell gonna move to the thymus. Once the immature T cell move to the thymus, they need to select the receptor. They need to select only good receptor. Firstly, they're gonna select the positive selection. So it means that the T cell need to weakly bind to the MHC molecule. Weakly, just only weakly. The reason is just because the T cell need to bind to the MHC molecule and also to the any other antigen. So the first step, they're gonna select the weakly binding MHC molecule receptor. And then the next one, they never ever bind to any other antigen. So it means that the T cell only bind to MSC, never ever bind to any other self antigen. This is a keyword of the T cell. So once you got the T cell that can bind weakly to the MSC molecule and never bind to any other self antigen, we're gonna call this as the mature or the naive T cell. The naive or the mature T cell gonna move out from the thymus to the secondary lymphoid organ. I mean, sometimes it's gonna be lymph node, sometimes it's gonna be spleen. The thing that happen is the T cell, sometimes they may go out to the blood circulation, but it's just about a recirculation, do nothing, never ever do anything, never do any function in the blood. It just go out and recirculation. So when your tissue got infection, when the situation occur, the dendritic cell gonna take some antigen. The dendritic cell takes some antigen, move from the site of infection with a lot of epitopes. This dendritic cell gonna move from the site of infection and go to the nearby lymph node with a lot of antigen. So the thing that happened is that the dendritic cell need to select just only one immunodominant to present on the MSC molecule. So the peptide presented on MSC molecule gonna activate T cell receptor at the first signal. And then this is a glue, the CD40 ligand and CD40 as a glue. But the second signal is when the B7 bind to the CD28 here gonna provide second signal. So this is a very important, the two signal presentation, the TCR by with the antigen on MSC molecule and also the co-stimulation is very important. At the end, if the activation is the MSC class 2 activate the CD4 T cell, you're gonna get T helper cell. If the activation of the MSC class 1 on antigen presenting cell activates CD8 T cell, you're gonna get cytotoxic T lymphocyte or the cytotoxic T cell. When you get a lot of effector cell, by the way, they're gonna have some memory occur. So some cell come to the effector cell, but some cell gonna turn to the memory along the way. So when you get the cytotoxic cell here or even a T helper cell here, these affected cell gonna move out to the site of infection. So the the T helper cell, they gonna secrete the cytokines. The T helper one, they gonna secrete cytokine to enhance the macrophage and also the enhance inflammation. So all about of the T helper one is all about enhance your immunity. T helper 2 is all about healing. T helper 2 is all about healing. And the T helper 17, they're gonna secrete the cytokine to call the PMN, I mean the neutrophil from the blood to the site of infection. So all the helper cell is all about the cytokine secretion. But about the cytotoxic T cell, the cytotoxic T cell gonna bind to the infected cell and try to kill those infected cell by two mechanisms, perforin with enzyme or the mechanism of the apoptosis induction using the fast and fast ligand. So once you got infection at the first time, I would say that in your blood circulation gonna have both memory and naive cells circulate in your body. But it's just circulation to transfer the cell from one side to the one side, one side to the one side, one side to the one side. Do nothing in the blood, but they just move from the one lymph node and transfer to one lymph node and transfer to another lymph node. It's just an exchanging of the cell in your body. That's it. So about the T cell, that's all about the T cell. If you guys have any question right now, please turn on your microphone and ask me if you have any question on the T cell. Any question now? Okay, it seems like no any question. But anyway, if you have any question, you may try to contact me by the line, by also by the email. I'm gonna give you the contact at the end. Or even you can post in the e-learning. I'm gonna come to the e-learning and also try to answer every question that you got. Okay, so next, I'm gonna give you a lecture for the next thing. Okay. Would you like to have some break? Okay, no, it seems like no. If no, I'm gonna go to the next part. So I just finished lecture on the T cell. There's a lot of things in the T cell. But next, I'm gonna give you a lecture about the B cell. So the B cell is quite complicated. It's quite complicated because normally the B cell, they need the activation of the T cell to help B cell again. So it's come from here. The B cell, normally the B cell, they need to wait for antigen come to them. It's unlike T cell. T cell have antigen presenting cell, take antigen and present to the T cell. But for the B cell, they just need to wait the antigen by themselves. So the source of antigen, it could be the circulation. Sometimes the antigen could come with the lymphatic vessel. You can see here there's a lot of vessel. There is a lot of vessel here. So the antigens that moving around, in Thai we call antigen is come from with น้ำเหลือง. So our serum, they have an antigen and also our serum gonna deliver antigen to the B cell. So once the B cell take, this is a picture. So once the B cell here, the B cell at your right hand side here, once the B cell recognize to the epitope, you need to pay your attention right now because this is thing that go through a very complicated things. So look at this. So the B cell, they recognize to the epitope here. This is a green one. It means that this B cell is specific to the green epitope here. This B cell specific to the green epitopes here. Okay, that's only the green epitopes. But when the B cell eat this antigen and process, and then the peptide that is the immunodominant gonna be the brown one. So it means that these B cells specific to the green antigen, but when they present the peptide on the surface, they present the peptide with the brown one. Sorry. They present the peptide with the brown one. So the B cell specific to the green antigen, but present the brown antigen is gonna move out from the B cell zone to the T cell zone to find any T cell that's specific to the brown antigen. Right now, B cell specific to the green antigen, but the T cell specific to the brown one. So if they can find each other, the T cell gonna be activated, and also the B cell gonna take some cytokine from the T cell. So the B cell at this stage, they gonna come to the plasma cell. So it means that the first step is mean that the B cell need to take antigen and then present the antigen to the T cell. If the T cell that's specific to the B cell, it's gonna help B cell differentiate into the short-lived plasma cell with some epitope, with some isotype switching. So it means that at this moment, you get the B cell, you get the T cell, and also you get the IgG, but this plasma cell can be died shortly. But anyway, if the bonding between these B cell and T cell is strong enough, they both of them gonna move together. Both of them gonna move together from the T cell zone and go into the germinal center. The germinal center is a marker that, okay, if you take some antigen or even if you take some vaccine and if you want to know you're gonna have a long memory or you're gonna have a really good response or not, we're gonna look if you got the germinal center. Right now, the T helper cell that go together, that go along with the B cell, once they come to the germinal center, they're gonna change the name. It means that they're gonna change the behavior. They're gonna change their lifestyle into the T follicular helper cell or the TFH. So if any B cell come with the TFH and from the germinal center, those B cell gonna have a high affinity because this TFH gonna help B cell to do affinity maturation. And also it's gonna have the isotype switching for sure. And if there is a germinal center, TFH gonna help the B cell to form the memory B cell. And also the plasma cell with the TFH activation, they're gonna be a long-lived plasma cells. So that is a principle. This is about, okay, when you need to imagine you activate the plasma cell in your lymph node. Yes, you're gonna have antibody secrete. But lymph node is a very small organ, and also normally plasma cell when they stay in the lymph node, they're gonna die fastly. So if you got the plasma cells, the plasma cell with the long life, they need to exchange. For example, if the plasma cell produce the IgG, the plasma cell produce the IgG, they're gonna move from the lymph node, move out, go to the blood, and then the IgG plasma cell gonna move to the bone marrow and stay in the bone marrow for a very, very, very long time. But if the plasma cell produces IgA, the plasma cell from the lymph node with the IgA gonna move to the blood and then come to the mucosal and stay secreting the IgA. So this is about the B cell activation and the migration. And then this B cell, you need to understand that not always the B cell with the antigen and also can with the T cell helper help the B cell can generate to the plasma cell is not always can find the T cell. So the B cell that can find the B cell is only because of the B cell can bind to the protein antigen because the T cell only recognize to the peptide. So we call this protein antigen as a T-dependent mechanism. So the B cell bind to the green epitopes here and then digest and then present the brown one to the helper T cell. You're going to get the memory cells. You're going to get a high affinity antibody. You're going to get a long-lived plasma cell because you can find the specific T cell to help. We call this protein antigen as a T dependent. And then if you expose to the polysaccharide antigen, this antigen could also activate the B cell to secrete antibody. But these polysaccharide have never been present to the T cell. The reason is just because of T cell recognize to the peptide present on MSC molecule. So the polysaccharide, the sugar, the lipid, anything that is not protein, gonna be only activation of B cell, not activate any T cell. So you're gonna get low affinity antibody and you're gonna get only IgM. And the plasma cell here is a short-lived plasma cell. It's gonna die very fast. And next, so there is an application because right now we know that if you got the antigen with the sugar, lipid, and also if you have the polysaccharide antigen, but anyway, you say that this antigen is so good, you want the antibody, you want a very long-term immune response to this polysaccharide, to this lipid, or even to this sugar. There is an application. You just easily conjugate this polysaccharide with protein. Because I also try to mention that these B cells specifically bind to polysaccharide. This B cell bind to polysaccharide and then they're gonna take polysaccharide with protein into the cells. And then this protein gonna be digest, and the protein gonna present to T cell. That's why I try to recall you that these B cells specific to polysaccharide, but when they digest the protein, they can present protein and activate T cell. This T cell is the carrier peptide specific T cell. It's gonna secrete cytokine and help B cell to produce antibody. So you're gonna get a long-term memory response, and also you're gonna get the IgG because the T cell can help antibody isotype switching. So talking about the effector B cell, this is different from the T cell. So the T cell, when they go to the effector cell, they need to move to the site of infection. But for the B cell, it's never ever move out. B cell only secrete antibody and send antibody out. So the antibody, if you can remember, the antibody can have the function of the complement activation. So the complement activation could do the function of the opsonization to make phagocytosis even better, faster, and more effective. The complement can activate the inflammation. And anyways, the last thing, if you activate the complement from the C1 to C, last thing, C10, you're gonna have the MAC, the membrane attack complex to lyse the microbe. So the first function is the antibody could activate the complement. The next thing, the antibody could bind directly to the toxin or even to the virus and make those viruses have no capacity to infect the cell. We call this is about a neutralization. So this is a directly binding and neutralize the function of the microbe or even the toxin. And then even antibody, when they bind on the pathogens, the phagocyte can detect this binding and can go to kill this pathogen even faster. We call the effect of antibody binding, and the phagocyte can do function even better. We call opsonization. And the last thing, antibody when they bind on the abnormal or on the infected cells, NK cell could detect the antibody bind on the cell and try to kill those cells. We call this is the antibody-dependent cellular cytotoxicity, or we call in short as the ADCC. So that's all about the B cell activation. So right now, I'm gonna come to the selection of the B cell, because you need to imagine right now, B cell activation, when they activate, they can activate a lot of cell. And once if the B cell have follicular T cell to help, if B cell have helper T cell to helper T cell here, the follicular helper T cell, gonna secrete the cytokines and let B cell edit the receptor all the time. Follicular T cell gonna let B cell edit receptor all the time. You need to imagine it's just like the B cell and T cell. The T cell is the center. The T cell is the center. If any B cell can come closer to the T cell, they're gonna get food. So it means that any high affinity B cell, they're gonna get food from the T cell or even from the dendritic cells. They're gonna get food, so they're gonna stay alive. Any B cell that edit receptor but lost binding. So it means that edit receptor but the affinity is just lower or lost the binding. They're gonna be kicked out from the T cell and they're gonna have no any food, no any cytokine. At the end, they're gonna die. We call the step that we try to select, only high affinity B cell and to get food from the dendritic cell and from the follicular T cell. We call this mechanism as the affinity maturation. The affinity maturation, because of the T cell, when they secrete the cytokines, the B cell gonna randomly mutate the gene. They're gonna randomly mutate the gene. So if you look at the upper one here, so at the first, yeah, it can bind to antigen, but low affinity. But when the T cell secrete the cytokine and let B cell randomly mutate and it comes to the higher affinity, this cell gonna have the higher affinity antibody production. We call this the mutation with your cell, with your somatic genome. We call mechanism as a somatic hypermaturation. So right now, I'm gonna give you the take-home message right now about the B cell. So the B cell is produced by the bone marrow, and even mature to the naive B cell in the bone marrow. That's why we call the B cell or the bone marrow cells. So the B cell or the bone marrow cell or the mature B cell gonna move from the bone marrow and go into the lymph node or spleen that we call the secondary lymphoid organ and stay still, wait for activation. The problem is that they just only waiting. So the antigen need to come to the secondary lymphoid organ and activate the B cell. So if the antigen is the lipid, carbohydrate, or even polysaccharide, or even a sugar, all of these things is gonna be T-independent antigen. So this B cell just recognize to those antigen and produce IgM. That's it. That's all. Nothing more. But if the antigen come to the lymph node with the T-dependent antigen, the T-dependent antigen is just only one thing. I mean, only protein. This T-dependent, the B cell gonna recognize to the epitopes, and then it's gonna present the peptide to the T cell. If they can find a specific T cell, this gonna develop into the plasma cell and have some switching, can produce IgG, but it's gonna have a short-lived plasma cell here. If this B cell with the T cell come both together come to the terminal center, and from the terminal center here, the T cell, helper T cell, gonna develop into the follicular helper cell or the TFH. This TFH gonna produce a lot of cytokine to activate the B cell. So the B cell can have the isotype switching here, can have the long-lived plasma cell here, and can have the memory. At the end, all the antibody just secrete and sent to the blood circulation to do function. But when they got a plasma cell here, if the plasma cell is the IgG. The plasma cell with the IgG gonna move to the bone marrow and keep staying, secrete IgG. If the IgA, the plasma cell with the IgA gonna move to the mucosal and stay secreting IgA. This is all about the B cell. Any question about the B cell right now? I guess not. Okay. Even though if you have any question, you just send me your email or anyways, I'm very happy to answer you. And the last lecture, I'm gonna lecture you about the phase of exposure. So the phase of exposure come with, you need to understand this is the first thing that happen in your body when you get some infection. So when you got the first time infection, you need to understand activation of the adaptive immunity. They take time. If you can remember, activation of T cell, they need two signal, and I would say that everything gonna be very, very slow. So once you get the pathogen here, antigen presenting cell gonna take some peptide and present the immunodominant peptide on MHC molecule. Activate T cell by activate through the T cell receptor as the first signal. The second signal comes after. So it take around 7 days. Same thing. The naive B cell, when they get antigen, when the B cell get antigen, they need time to activate themselves and going to the antibody secreting cell. I would say that this effect take 7 days. Along the way, when they activation, they're gonna expand themselves. The expansion is mean increase in number. The T cell and B cell need to increase in number. Some of the cell go into the effector cell, but some of the cell go into memory cell. They just divided themselves, divided. Along the way here, when you take the antigen at the first time, I would say that your immune response gonna peak. I mean, stay at the highest concentration around 7 more days. So it means that if you want to see a very first response, you should take a blood at 7 days. If you want to observe the later immune response, you should take antibody at 14 days. After 14 days, I would say that all the immune response gonna be decline phase. It's gonna drop down here, but not completely disappear. They gonna keep staying cell or the surviving memory as the memory cells here. So start from zero, but at the end they keep something. This is the first immune response. So it's come to the application. If you want to know, okay, you're taking the blood, for example, if you want to test some vaccine, you inject vaccine to people. Before injection, you need to keep the blood at the day 0. And if you want to know the vaccine priming or activation occur or not, the first activation gonna occur at the 7 days. And then if you say that you want to see the antibody at the highest concentration in your body, you should take the blood during 7 or 17 days here. And then if you want to know, okay, if you want to see the the memory cells, you should take the blood at more than 21 day, 21st day. So this is what the application that why you need to know the phase of exposure. If you want to design your experiment, you may try to keep the blood at the week 0, the first week, the second week, and then the third week with different purpose. So that's all about the first exposure. So it's come to the secondary exposure. So you need to understand this is the first exposure here. You start with 0, but you end with some degree of immune response. You should take around maybe 4 weeks. No, sorry. You should take around 2 weeks to activate. But if you get the secondary infection or you expose to the to the same antigen for the second time, so the response here you can see that the response from maybe the week number 5 here to the peak one. Normally, when you want to peak immune response, you should take 2 weeks. But if you expose to the secondary time, the response gonna be immediately. So it means that it skip all the priming step. Your immune response gonna be keep going so fast and even higher than the first immune response. So this is about a secondary immune response. And if you pass through the secondary immune response, you're gonna have more, even more memory cells. This is the reason why when you take some vaccine, you take for the first shot, you wait for a while, you're waiting for what? You're waiting for your immune system to come to the normal state and to be ready to respond to some new thing. And then you're getting the booster dose for the second shot. So the booster shot dose gonna activate your immune response more stronger, and at the end, you're gonna have more memory immune response at the end. So I would say that should be all of my lecture for today. I'm sorry I can't join you guys in class, but anyway, I think I try my best to explain everything as much as I can. So lecture today, I summarize from these two textbooks. So if you want to learn, you can learn. I guess Ajahn Visisak have taught you how to go to the critical key and download this textbook. So if you want to learn, if you want to read it, just go to the critical key and download this textbook. I would say that this textbook is the master key for every immunologist. So last time and last chance, if you have any question, please turn on your microphone and ask me right now.
อาจารย์คะ
Okay, no. If no any question, I would say that my lecture, if you have any question, you may try to email me, and even you can add the line and ask me the question, or even post to the forum in the e-learning, or even ask in the line group. I would say that I'm very happy to answer every question. So นักศึกษาไทยอยากได้ภาษาไทยไหมคลิป For Thai student, I'm gonna summarize with the Thai language again. Sorry for the international student, but for Thai people, I'm gonna summarize it again with Thai version. วันนี้สิ่งที่อาจารย์อยากจะเน้นนะ I would say that the take home message, I would say that my examination is just only in the slide of take home message. Okay, so right now I have one more hour. I'm gonna tell you everything about my examination. So please take a note. And I would say that if you understand this, you can do my examination. So my examination, you need to go and look for the T cell immune response if you want to recall the knowledge. I would say that if you can explain this take home message, I would say that you can do my examination. So I'm gonna ask you about how about the selection here. So please take a look about the selection here. Please take a look. And also I'm gonna ask you about the function of T helper cells. What is the function of T helper cell? What is the function of the cytotoxic T cell? I'm gonna ask you this. So please remind about this thing when you prepare your examination. I would say that for the T cell, that's it. The production of T cell produced by the bone marrow, but mature in the thymus. I would say this is the first point important. You need to make sure that you can remember this. And then the selection of the T cell is the weekly select by the MSC, select by weekly by to MSC, but never ever bind to the self antigen. This is the second point. You need to go back and take some remind. And once you get the memory cell or anything here, you should know about the T helper cell function is about the secreting cell, the secreting cytokine. You need to know about the cytotoxic T cell, about the killing of the abnormal cell. That's it about the T cell. And my examination, another examination of the B cell, is just the take-home message as well. So the B cell here, I'm gonna ask you just only, you need to know B cell produce and mature in the bone marrow. And the selection of the B cell is just never ever bind to your self antigen, never ever. And also the second thing that you should know is about the T dependent. The T independent is mean the lipid, the carbohydrate. Please remember, if you take any carbohydrate or lipid, you're gonna activate only IgM. carbohydrate and lipid is the T independent, you're gonna activate only IgM. But if you take protein antigen, you're gonna activate the class switching, you're gonna activate the plasma cell activation, you're gonna activate the memory. Okay, so if you take the protein, you can have IgG because of the class switching. You can have the plasma cell with the long live and also the memory. If you can't remember the long live, please remember IgG and memory, please. And yeah, I would say that my examination gonna be like that. And I gonna keep you guys explanation in Thai because I know that your Thai student may can't follow me something อันนี้ขอเป็นภาษาไทยอีกรอบหนึ่งนะสำหรับทีเซลนะสิ่งที่อาจารย์เน้นเลยก็คืออยากให้รู้ว่ามันสร้างมาจากไหนให้เรารู้ว่าทุกเม็ดเลือดในกระแสเลือดเราสร้างมาจากโบนแมโรแต่พิเศษก็คือทีเซลสร้างที่โบนแมโรมันจะได้แค่ตัวไม่เต็มวัยหรือที่เรียกว่า immature T cell immature T cell เขาจะเดินทางไปยังที่ไทมัสนี่แหละคือเหตุผลว่าทำไมเราเรียกว่า T cell เพราะมันโตและโตเต็มวัยที่ไทมัสที่ไทมัสเขาทำอะไรบ้างเขาจะทำอยู่สองอย่างคือหนึ่งเลือกเซลล์ที่จับกับ MSC โมเลกุลได้สาเหตุผลก็ตามที่อาจารย์บรรยายไปว่าเพราะว่า T เซลล์มันมีสองขยักที่หนึ่งมันจับกับ MSC เพราะว่าเปปไทด์ที่นำเสนอจะต้องเป็นเปปไทด์บน MSC ดังนั้นสเต็ปที่หนึ่งเขาเลยต้องเลือกเซลล์ที่จับกับ MSC โมเลกุลอ่อนๆได้ตรงนี้ย้ำออกข้อสอบชัวร์อันที่สองเขาต้องไม่จับกับแอนติเจนอื่นๆเลยถ้าเอาแอนติเจนหัวสมองให้ต้องไม่จับเอาแอนติเจนหัวใจให้ต้องไม่จับเอาแอนติเจนตับไตไส้พุงให้ต้องไม่จับดังนั้นห้ามจับกับแอนติเจนอื่นๆเลยนี่คือคอนเซปต์ในการทำให้ไทมัสกลายเป็นตัวเต็มวัยเมื่อไทมัสเป็นตัวเต็มวัยเมื่อไหร่ปุ๊บตัวเต็มวัยเขาจะเดินทางออกจากไทมัสไปเก็บเอาไว้แล้วรอการทำงานการรอการทำงานจะรอไว้ที่ลิมโนดหรือสปลีนเพราะฉะนั้นลิมโนดกับสปลีนเนี่ยเขาเรียกรวมๆว่าเป็น secondary lymphoid organ secondary lymphoid organ เขาจะแค่เก็บเซลล์ไว้เฉยๆคราวเนี่ยเซลล์อ่ะมันจะมีการเดินทางแลกเปลี่ยนกันด้วยเพราะอะไรเขาไม่ได้เดินออกไปในเลือดเพื่อเอาไปทำงานเขาเข้าไปอยู่ในเลือดเพื่อที่จะแลกเปลี่ยนลิมโนดลิมโฟไซต์หรือว่าแลกเปลี่ยนทีเซลล์ระหว่างลิมโนดระหว่างสปลีนเขาเข้าไปสู่เลือดเพื่อเดินทางรอบตัวเราเขาไม่ได้เดินทางเพื่อไปทำงานเพราะฉะนั้น naive T cell mature T cell เขาจะมีการเข้าสู่บัตร circulation เพื่อเดินทางเฉยๆเมื่อไหร่ก็ตามเกิดสถานการณ์มีการติดเชื้อในทิชชูให้เราเข้าใจว่าเมื่อมีการติดเชื้อเสร็จปุ๊บ dendritic cell จะไปทำการเก็บแอนติเจนมาแอนติเจนต่างๆนานาตัวนี้จะเน้นเสมอว่าแอนติเจนที่เราเก็บมาหนึ่งครั้งหนึ่งโปรตีนอ่ะมันมีหลายอีพิโทปมากๆไอ้เจ้าเดนเดติกเซลจะทำการย่อยโปรตีนกลายเป็นชิ้นส่วนเล็กๆเล็กๆเล็กๆเปปไทด์เล็กๆเต็มไปหมดเลยไอ้เจ้าตัวนี้แหละเดนเดติกเซลจะเดินทางไปยังลิมโนดเดินทางจากที่ติดเชื้อเข้าไปสู่ลิมโนดที่ใกล้ที่สุดการทำงานก็คือเดนเดติกเซลจะเลือกเอาเปปไทด์ที่จับกับ MSC ได้ดีที่สุดขึ้นมานำเสนอไอ้เปปไทด์ที่จับกับ MSC ได้ดีที่สุดนี่แหละเขาเรียกเป็นอิมมูโนโดมิแนนต์เปปไทด์ที่จับได้ดีที่สุดอิมมูโนโดมิแนนต์จะถูกนำเสนอผ่าน MSC โมเลกุลแล้วจับกับ T cell receptor นี่คือสัญญาณที่หนึ่งสัญญาณที่หนึ่งอย่างเดียวมันแรงไม่พอถ้ามีแค่สัญญาณเดียวจะเกิดเหตุการณ์ energy ก็คือพลังงานไม่พอดังนั้นมันต้องเกิดสัญญาณที่สองด้วยแต่สัญญาณที่สองจะเกิดขึ้นได้ก็ต่อเมื่อเซลล์มันเข้ามาใกล้กันมากพอดังนั้น T cell เขาเลยต้องสร้าง CD40 ไลแกนมาจากกับ CD40 แล้วสัญญาณที่สองถึงจะเกิดขึ้นเมื่อมีสัญญาณครบทั้งสองสัญญาณเขาจะทำการพัฒนาจาก naive B cell จาก naive T cell ไปเป็น effector cell effector cell ก็คือถ้าสัญญาลักษณ์บน T cell เป็น CD4 หรือ CD4 แล้วนำเสนอแอนติเจนผ่าน MC class 2จะได้ T helper cell ถ้าสัญลักษณ์บนเซลล์เป็น CD8 นำเสนอแอนติเจนผ่าน MC class 1จะได้ cytotoxic T cell คราวนี้เวลาที่เซลล์มันแบ่งตัวมันจะมีทั้งส่วนที่แบ่งตัวออกมาแล้วกลายเป็นเซลล์เอาไว้ทำงานเราเรียกว่า effector cell กับเซลล์ที่แบ่งเอาไว้เฉยๆแล้วรอเก็บเป็นความจำเรียกว่าเป็น memory มันไม่มีอะไรบอกว่าเซลล์ไหนจะเป็นอะไรมันจะเป็นการสุ่มเลยส่วนหนึ่งแค่ส่วนน้อยนั้นที่เก็บเอาไว้เป็น memory ส่วนใหญ่จะเป็น effector cell effector cell ทั้ง T helper ทั้ง cytotoxic T cell เขาจะถูกปล่อยไปยังที่ที่มีการติดเชื้อการทำงานก็คือถ้าเป็น helper cell เขาจะสร้างไซโตไคน์อย่างที่อาจารย์เกริ่นไว้ตอนต้นก็คือ T helper cell ทุกวันนี้แบ่งออกเป็นหลายคลาสมากๆหลายสับไทป์มากๆแต่ทุกอันหน้าที่เหมือนกันคือเอาไว้สร้างไซโตไคน์เอาไว้ควบคุมการทำงานของภูมิคุ้มกัน T helper 1ให้จำเอาไว้เลยว่าการทำงานของร่างกายมนุษย์นะ1มาก่อน2เสมอเมื่อมีการติดเชื้อ T helper 1จะสร้างไซโตไคน์เอาไว้กระตุ้นการทำงานของภูมิคุ้มกันทั้งกระตุ้นเม็ดเลือดขาวกระตุ้นการอักเสบแล้ว T helper 2จะตามมาทันทีเมื่อมีการอักเสบเมื่อมีอะไรก็ตามแต่ร่างกายต้องการการสมานแผล T helper 2จะเกิดขึ้นเพื่อเอามาสมานแผลคราวนี้ T helper 17อย่างที่บอกเห็นไหมว่า T helper 1ก็คือ T helper 1นั่นแหละ T helper 17มันมีเลข1ขึ้นเหมือนกันดังนั้นฟังก์ชันเขาจะคล้ายกันคือเอาไว้ตอบสนองต่อการติดเชื้ออะไรบางอย่างแต่ T helper 17เขาจะทำหน้าที่พิเศษขึ้นมาหน่อยก็คือ IL-17มันจะเอาไว้เรียกระดมพลนิวโตฟิลเพราะฉะนั้นตอนนี้จะเข้าใจแล้วเนาะว่านิวโตฟิลที่อยู่ในเลือดพวกเนี้ยมันจะเข้ามายังจุดที่เกิดการติดเชื้อได้ไวมากขึ้นได้ดีมากขึ้นได้เยอะมากขึ้นเมื่อมี T helper 17แล้วสร้าง IL-17ใดๆก็ตามแต่ตอนนี้ T helper 1 2 17เอาไว้สร้างไซโตไคน์ส่วนไซโตท็อกซิก T เซลล์เขาจะมีหน้าที่เอาไว้ทำลายเซลล์ที่ผิดปกติไม่ว่าจะเป็นเซลล์ติดเชื้อเซลล์ที่เป็นมะเร็งหรือเซลล์ที่มีการบาดเจ็บการทำลายเซลล์ที่บาดเจ็บทั้งหลายทั้งแหล่พวกนี้จะมีอยู่สองกลไกใหญ่ๆจะใช้กลไกไหนก็ได้แล้วแต่เซลล์กลไกที่หนึ่งคือใช้เปอร์ฟอรินกับแกลนซามเปอร์ฟอรินคือเอาไว้เพิ่มการเพิ่มแปลว่าการทำรูเมื่อไหร่ก็ตามเซลล์มีรูเสร็จปุ๊บเขาจะฉีดแกนไซม์เข้าไปแกนไซม์ทำให้เซลล์เข้าสู่กระบวนการอะปอปโตซิสแล้วก็ตายอีกกลไกหนึ่งคือเป็นการ induce ผ่าน fast และ fast ligand เขาเรียกเป็นอะปอปโตซิสนี่แหละเพราะฉะนั้นใดๆก็ตามแต่ให้รู้ว่าถ้าเป็นไซโตท็อกซิก T เซลล์เอาไว้ฆ่าเซลล์ผิดปกติเนาะคราวนี้ให้เรารู้ว่าเมื่อไหร่ก็ตามที่เราติดเชื้อเสร็จแล้วสิ่งที่มันจะเกิดขึ้นก็คือนี่แหละเราจะได้เมมโมรีแล้วก็ได้นาอีฟเซลล์เซอร์คูลเลตการเซอร์คูลเลตไม่ได้เอาไว้ทำงานเอาไว้แลกเปลี่ยนเซลล์ระหว่าง secondary lymphoid organ เท่านั้นอันนี้คือ take home message ของ T cell เพราะฉะนั้นเดี๋ยวจะให้ take home message อีกครั้งหนึ่งของ B cell แต่เป็นเวอร์ชันภาษาไทยสำหรับ B cell แล้วมันจะซับซ้อนหน่อยเพราะว่า B cell เขาต้องกระตุ้นหลายขั้น B เซลล์เริ่มต้นเลยก็คือ B เซลล์เขาสร้างและพัฒนาตัวเต็มวัยที่ไขกระดูกการที่เขาอยู่ที่ไขกระดูกนี่แหละเขาเลยเรียกว่าเป็น B เซลล์หรือ bone marrow cell เนาะเมื่อไหร่ก็ตามที่เขาทำเป็นไขกระดูกโตเต็มวัยให้จำเอาไว้อย่างเดียวว่า B เซลล์จะไม่จับกับเซลฟ์แอนติเจนเลยเด็ดขาดไม่มีข้อยกเว้นถ้าจับเมื่อไหร่ต้องไปเปลี่ยนรีเซพเตอร์ถ้า B เซลล์ที่ทำงานได้ดีเขาจะส่งเข้าสู่กระแสเลือดแล้ว B เซลล์จะทำหน้าที่แค่รอแอนติเจนเข้ามาเท่านั้นเขาจะรอแอนติเจนอย่างเดียวเลยในลิมฟ์โนดเมื่อไหร่ก็ตามที่มีแอนติเจนเข้ามาตัวอย่างที่หนึ่งก็คือถ้าเป็นแอนติเจนที่เป็น T independent ก็คือไม่ขึ้นกับ T เซลล์เลยได้แก่แอนติเจนประเภทลิปิดคาร์โบไฮเดรตลิปิดกับคาร์โบไฮเดรตจะกระตุ้น B เซลล์ได้แล้วได้แค่ IgM อย่างเดียวไม่ได้อย่างอื่นออกข้อสอบพูดเลยอันถัดมา B เซลล์ที่กระตุ้นด้วยแอนติเจนที่เป็น T dependent แปลเป็นไทยก็คือ T แอนติเจนที่ต้องมี T เซลล์เข้ามาช่วยอาจารย์จะพูดเสมอว่าให้ดูภาพนี้ดีๆนะ B เซลล์ตัวนี้เขาจับกับสีเขียวนะมัน specific กับ antibody specific กับสีเขียวแต่เมื่อจับเสร็จปุ๊บเขาจะดึงแอนติเจนเข้ามาในเซลล์ย่อยเป็นชิ้นเล็กๆๆๆๆๆๆแล้วคัดเลือกอิมมูโนโดมิแนนต์ปรากฏว่าอิมมูโนโดมิแนนต์มันเป็นคนละส่วนนะเขานำเสนอตัวสีน้ำตาลสเปกซิฟิกกับเขียวแต่นำเสนอตัวสีน้ำตาลให้กับ T เซลล์นั่นหมายความว่า T เซลล์อ่ะเป็นตัวจำเพาะกับสีน้ำตาล B เซลล์จำเพาะกับแอนติเจนสีเขียวตรงนี้คือคอนเซปต์ใหญ่ที่ต้องรู้เนาะเพราะฉะนั้นเมื่อเรารู้ว่าโอเคเมื่อ B เซลล์ได้รับโปรตีนเปปไทด์เข้ามาเขาก็จะเอาไปนำเสนอให้กับ T เซลล์สุดท้ายถ้าเป็นการเสนอขั้นต้นยังไม่เดินไปไหนก็คือนำเสนอกันเสร็จปุ๊บจุ๊บกันเสร็จปุ๊บออกไซโตไคน์มาน้องจะได้ไอโซไทป์สวิตชิ่งสร้าง IgG ได้แค่นั้นแต่ถ้า T เซลล์และ B เซลล์ของคนนี้เขาตกลงปรงใจกันแล้วเขาเดินทางเข้าสู่ Germinal Center T เซลล์ตัวนี้จะเปลี่ยนชื่อเรียกใหม่เพราะมันไปอยู่ใน Germinal Center เขาเรียกว่าเป็น T follicular helper cell หรือ TFH TFH จะคอยสร้างอาหารตลอดเวลา การที่เขาสร้างอาหารตลอดเวลาทำให้ B เซลล์พวกนี้ได้รับการกระตุ้นตลอดเวลาเขาเลยเกิดสวิตชิ่งต่อไปแล้ว B เซลล์ที่เกิดขึ้นพลาสมาเซลล์ที่เกิดขึ้นจะมีอายุนานขึ้นเพราะมันได้รับอาหารนานขึ้นสุดท้ายจะเกิดเมมโมรีตรงนี้เพราะว่ามันได้อาหารจาก follicular T cell ดังนั้นตรงนี้ถ้าเราเข้าใจก็คือเราจะรู้ว่า affinity maturation มันเกิดขึ้นเพราะว่าเซลล์พวกนี้มันเกิดได้รับการไซโตไคน์กระตุ้นเรื่อยๆน้องจะเกิดมิวเทชันเรื่อยๆเรื่อยๆเรื่อยๆตัวไหนที่มิวเตตแล้วจับได้แย่ลงจะถูกถีบออกจากวงขาดอาหารตายตัวไหนที่มิวเตตแล้วเข้าสู่หรือว่าจับกับทีเซลได้แน่นขึ้นจับกับอาหารได้แน่นขึ้นน้องจะได้รับอาหารมากกว่าชาวบ้านชาวช่องแล้วน้องจะโตขึ้นเรื่อยๆแล้วน้องจะมีจำนวนมากขึ้นนั่นแหละเรียกว่า affinity maturation มันเป็นเหตุผลด้วยว่าทำไมแอนติบอดีเราเมื่ออายุมากขึ้น affinity มันก็จะดีขึ้นเพราะมันเกิด affinity maturation ใดๆก็ตามแต่ตอนนี้ให้เราเข้าใจว่าโอเคการตอบสนองตอนนี้นะเราสร้างแอนติบอดีแล้วแอนติบอดีจะถูกปล่อยเข้าสู่กระแสเลือดนี่คือข้อแตกต่าง B เซลล์เขาจะไม่ปล่อยเซลล์ออกไปข้างนอกเท่าไหร่ถ้าได้พลาสมาเซลล์แล้วแอนติบอดีปล่อยออกไปเท่านั้นไม่ปล่อยพลาสมาเซลล์ออกไปเพราะฉะนั้นเอฟเฟกเตอร์ที่ไปทำงานณจุดที่ติดเชื้อจริงๆคือแอนติบอดีคราวนี้มันจะมีอีกนิดนึงแอนติบอดีพวกนี้พลาสมาเซลล์พวกนี้ถ้ามันอยู่ในลิมโนดน้องจะอายุสั้นจริงอยู่ว่ามีเฮลเปอร์ชอยช่วยแต่ว่ามันจะยังไม่อยู่ยาวนานขนาดนั้นแล้วมันจะทำงานไม่ค่อยตรงจุดเท่าไหร่ถ้าเมื่อไหร่ก็ตามแอนติบอดีเป็นชนิด IgG พลาสมาเซลล์เป็นตัวสร้าง IgG น้องจะเลื่อนแล้วก็ขยับตัวออกไปถ้าเป็น IgG นะน้องจะเลื่อนและขยับตัวไปเก็บไว้ที่โบนแมโลทุกครั้งที่โบนแมโลเขาสร้างเม็ดเลือดทุกครั้งที่พลาสมาเซลล์สร้างแอนติบอดีมันก็จะปล่อยเข้าสู่เลือดพร้อมกันไปเลยเพราะฉะนั้นมันจะเป็นการเอาไปเก็บไว้ทำหน้าที่โดยตรงถ้าเป็นแอนติบอดีที่สร้าง IgA น้องจะเดินทางออกจากลิมโนดแล้วไปอยู่ตามเยื่อเมือกต่างๆเพราะว่าเยื่อเมือกต้องการ IgA ดังนั้นน้องก็จะไปอยู่ตามเยื่อเมือกแล้วก็สร้าง IgA ออกมาเรื่อยๆเรื่อยๆเรื่อยๆมันเป็นการอยู่ให้ถูกที่แล้วสร้างให้ถูกอย่างเนาะเพราะฉะนั้น B เซลมีคอนเซปต์เท่านี้ข้อสอบอาจารย์ออกเท่านี้เลยแต่ไม่ต้องห่วงเดี๋ยวก่อนสอบเดี๋ยวจะติวเรื่องนี้ให้อีกรอบนึงแต่ว่าใดๆก็ตามแต่เดี๋ยวจะโพสต์คลิปวันนี้ so after this class I gonna post this record into my e-learning so you can go back and check everything that I told you here every examination I told you here you can go back and listen again but don't be panic so in the Friday after the group discussion, I gonna remind you guys again. I try to summarize everything about the immunology for you guys. So you're gonna I believe you can do the examination so easy. So don't be panic. But anyway, do your job. So when you learn something, when you go back to your home, please recall your knowledge. But don't be panic. We try to help you out. So any more question? If no any more question right now, I gonna finish my lecture today. So I gonna collect the score by the examination or of my point here. And in the afternoon, Ajahn Suprani gonna give you a lecture in the lecture room. So please go to the lecture room. And I guess she have some score with the pre-test. And she gonna take a score in the class. So don't miss the class. Ajahn Suprani has no any final examination. So don't miss the class. Don't miss the class. Please go to the class. So if you have no any question right now, my lecture is end today, so see you maybe in the Wednesday. See you there. Okay, it's finished for today. Thank you very much, everyone. So you can leave the Zoom now. Bye bye.
อาจารย์ค่ะควิซนี่ไม่ต้องทำใช่ไหมคะหรือว่ายังไงที่มันอยู่ใน e-learning อะค่ะอาจารย์ค่ะไม่ได้ยิน
ฮัลโหลได้ยินไหม
ได้ยินแล้วค่ะ
อุ้ยทั้งหมดที่อาจารย์เลคเชอร์ไปเมื่อกี้มันได้ยินไหมนี่
ได้ยินค่ะได้ยินค่ะแต่
โอเค
เมื่อกี้ไม่
ควิใน
ได้ยิน
e-learning เป็น practice เฉยๆไม่เก็บคะแนน
แค่แค่ทำอันนี้เฉยๆใช่ไหมคะ
ใช่ใช่เป็น practice เฉยๆไม่เก็บคะแนน
อ๋อค่ะขอบคุณค่ะ
ครับโอเค